A very warm, a very warm good morning to all of you. So today I am pleased to introduce Sanjay J. Jambhulkar sir. He is head Nuclear Agriculture and Biotechnology Division, BARC Mumbai. He is alumni of IRI. He did his MSc and PhD from this premier institute itself. He joined BARC in 1991 and he has a very vast experience of working with development of mutants, especially in crops like sunflower, brassica, and then other wheat and other cereal crops. He won various awards at different arena. He is an illustrious scientist having a big list of awards from international and national institutes and society. He has published more than 100 research papers in different area of mutation biology. He has very good publications, especially in the area of novel mutation identification in sunflower, Indian mustard, and rice. And he developed many improved genotypes also using the tool of mutation. So with this brief note, I request my coordinator that to welcome today's invitee speaker. So today, sir,
Yeah.
So today, sir, is going to give a talk on induced mutagenesis for crop improvement. So now I request, sir, to please continue.
So good morning, everybody, and thank you very much to Dr. Ranjit Ranjan and his team to invite me to deliver a lecture on the mutation breeding. So I will start my lecture. Is it audible there? Yeah. So Bhabha Atomic Research Center, it's a premier institute in the field of nuclear sciences in India and in the world. And we deal with the use of radiations and the radioisotopes in the various aspects, like industry, medical, and even the national security and other things. And one of the major application is in the agriculture also. So we try to prove that generally the people know that the nuclear energy is for the some kind of the devastations and all these things. But there is a lot of applications that we say as a peaceful use of the nuclear energy. And in one of the area is agriculture. So we work on the nuclear agriculture and biotechnology division for the crop improvement program. That is a major one. But beside that, we have the application in the soil science, application in the entomology, application in the tissue culture, and even the food preservations. But this part I am not going to deal with. I will try to concentrate only on the mutation breeding. Hopefully, my time is, Ranjit, 1 hour? Yeah. Okay. So I will try to complete. What I will try to do, I will try to give you the example rather than going to the details of the techniques, because you know the techniques even up to the genomics. Okay? So I will not go into the basic of that, but whatever we have done, whatever world has done, so that I will try to give the example. This is the, all of you are well aware that our present yield is a 282 million tons, and what we need is in the future, it's a very high demand, that is 333 million tons. Whatever we got so far, it's mainly it is a product of the Green Revolution, and the Green Revolution happened due to the dwarfing genes, and Dr. Swaminathan is a father of Green Revolution in India, and he got that World Food Prize. But the reason for that is a isolation of the natural dwarfing mutants. And that is called as the spontaneous mutations. That is SD1 in the rice and RST1 and RST2 in a wheat. These are the spontaneous mutations. And in the world has got the whatever production has increased in the world and India, it is basically because of these genes. This germplasm was used in the crossing program. The world has used this, and whatever varieties are there, it is out of that. So suppose I say we are getting the food due to the mutations. Had these mutations would have not been isolated in the nature, probably would have not got this kind of the jump in the yield. So if I say that we are living or we are getting the food because of the mutations, so hopefully I am not wrong. And I feel that you will be also agree with me. So that is the magic of the mutation. That is the magic of the mutations. And the mutation, all of us, all living organisms on this globe, it is evolved through the mutations, whether it's animals, plants, microbes, and that evolution is still going on. But we do not know what kind of the mutations are occurring with me or in the human being or in the plants. So evolution is mainly due to the mutation, and in the plants, then it is a recombination. That is what the magic of the mutations. This is the, though we say that we are self-sufficient in the food grain production, 282 million tons. Again, in the coming year, it will increase. But the present situation of the GHI, Global Hunger Index, it is not that happy for the India. Whether it's a nutritional deficiency or it is availability of the food as such to feed the number of the stomachs in the, and this is, I feel it's a very grim situation. Out of 117 countries, it is not. Our neighboring countries, small countries, they are good. But our position is not good. So there now the younger generation should take over, and how to fill this gap, how the rank will come up to the low level, which will be good for the India. That is what there is a big things to work on. Yeah, this is the situation. If you remember the Bengal famine, so the things are goes, the million of people dies. That is what that is the importance of the food. And the philosopher like a Seneca says, the hungry persons listens neither to reason nor religion or nor bent by prayer. So just to experience yourself by keeping yourself hungry for the 5 days, and on the 6th day, if you ask that, okay, you get fresh, put your prayer, and then you eat. You will say you forget about all. First of all, you give me the food. But that needs the experience that you can see also. The challenges, everybody knows that the population, climate, the soil fertility, all these are the big challenges. And the solutions are, first of all, developing the high-yielding varieties. And with the recent trends of the climate, says new diseases, the new abiotic stresses, and therefore the adaptation of the variety to climate change. And this is what, again, if you see the nutritional hunger point of view, we have increased our yield. But if you see the quality of the high-yielding varieties or the nutritional composition of the high-yielding varieties compared to our old germplasm or the old varieties, it is not that good. And therefore, I think this particular training program is for that. So that is the major challenges. Now, what has happened so far, it is up to 218 or 19. This was okay. We are getting the food. We are producing the high-yielding varieties. We are managing the population also. So from productivity to the getting the food by each and every person, it was okay. But now onwards, this gap has started, and it will increase in the. This is going to be food demand. and this is what the food supply in the future. So therefore, increasing the productivity, it's a per unit area that will be the major challenge by avoiding the loss and avoiding the food supply. This is what the major challenge. So this should be narrowed down in the future. And therefore, our developing the high yielding varieties with the better nutritional quality will be the major challenges for all of you and all of us. So far, we were doing the conventional plant breeding, and we have reached to this stage, including some of the biotechnological aspects. Okay, but now the things like various omics technologies with the it's like CRISPR and tilling is going to play a major role for the modification of the crop plants. But beside this traditional mutation breeding and the omics or the biotechnological aspects, I emphasize on the mutation breeding. You ask me why? Why the mutation breeding? Now, suppose if you take any crop, say like a wheat, rice, or in that case any pulses or any oil seeds, you find that the yield potential of all these crops has reached to the particular stage. Say like a wheat, suppose we are getting 60 quintals per hectare. With our efforts, it will go to the 70 quintals per hectare, 80 quintals per hectare, and now we are expecting 100 quintals per hectare. Okay, say like rice also. So I was in the BISA some 2 years before, and they put all varieties in a row. The released varieties since 1950s to 1918. And I found there is hardly any difference. Okay, hardly any difference. But still, every year we release the variety showing that it's a 5% increase, 10% increase, and all these things. Now, suppose I have the plant which is giving the, say, 70 quintals in a farmer's field in that case. Okay? Our experimental may go up to the 100 quintals. Now, above this, suppose you want a yield increase. What is the solution? The first solution, unless and until you have the variability for the particular character or the range of the character, which so far in the last 50 years, people have exploited or the scientists have exploited the all germplasm. You have done all possible crosses and come to the stage of the 70 quintals per hectare. Now, suppose if you want to go beyond this, unless and until you have the variability for the particular trait, the challenges which I put it over there for that particular trace, yield and yield contributing character, resistant to biotic and abiotic stresses, novelty in the nutritional character, then and then there is a possibility to go ahead. Otherwise, using the two high yielding varieties, we have the experience, there is hardly chance of increasing the yield potential. Suppose there is a one variety of 70 quintals per hectare, another is a 75 quintals per hectare. I cross these varieties and try to isolate a recombinant which will be the 80 quintals per hectare. The chances is very less because I have put all the possible good genes in the one variety. Therefore, it is giving the 70 quintals per hectare. Another variety which is giving the 75 quintal per hectare, there also I tried to put all possible good genes. It means that they have come to the saturation level. In the genomics point of view or the gene point of view, when all the things are better, so when you cross it, what is the chance of getting the recombinants which will be 80 quintals per hectare? It's a very less, very less. So unless and until you modify the genome, say maybe the biochemical pathway, maybe the physiological pathway, or the anything which will try to provide you the new kind of the recombinants, then and then. I will give some of the examples how the mutations have broken the yield barrier of the particular crop. And therefore, unless and until we have the new germplasm in our hand, it's very difficult, though we'll claim that we want to develop a variety which will be 100 quintals per hectare. In my opinion, it will be very, 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 very difficult, unless and until you generate the new germplasm. Or else, you try, if you don't want to do it, then you exploit the all germplasm available in the world, which people have done. Our seniors or those who have worked for the crop improvement, they have slogged their life to develop the high yielding varieties by using the existing high yielding germplasm. And therefore, so there is now, we should go ahead with the new ideas. New ideas is unless and until you have the new germplasm in your hand, it will be very, very difficult. Okay? So therefore, I feel the changing the germplasm or isolating the new germplasm, mutations is the only solutions. There is another possibility of using the wild germplasm. But when you use the wild germplasm in the crossing program, you will find a large number of the genetic drags. Unwanted genes comes in there. And to delete that, again it is we have seen in the brassica, Dr. Banga has done a lot of work on that. Even IRI has done a lot of work on this. But in this process, what happens, we have developed the plant, which will have the particular character, but it is not showing the high yielding potentiality. And sometimes the undesirable characters are also getting involved. Now to delete this, again, we'll have to do the mutation. Okay? Unless and until you delete that, and that is again the long process. So in the mutation breeding, what happens? You take a high yielding varieties, okay? Okay, before that, I will see this is the general applications of the nuclear energy you can do for the pest control. This is our main objectives: food and environmental protection, soil, water, and animal protection. If you really want to go for the details of the application of the nuclear techniques in the agriculture, now this is what I was talking about, that there is a need to strengthen the germplasm. Now, how the crop evolved? If you see the evolution of the crop plants, say maize, it was a teosinte like this, and now we have the maize, it is like this. It is nothing but the mutation. The mutation and the recombinations. Now, just to think of the maize crop looking like this, and now the big crop like this, and just to say within the hybridization program would have taken a long years too, because there was the mutations, good mutations, and that we used in the crossing program. And likewise, even in the rice or the wheat or even the tomato, if you see, even the sunflower. So if you look in the history of the evolution of all the crop plants, they are the product of the mutations. They are the product. And the very well examples which I told in the wheat and the rice. So without mutations, it's very, very difficult to go ahead. Now, why the mutations again? So this is what the spontaneous mutation, their frequency is in the 1 lakh or the 10 lakhs, you get it. But in the induced mutations, that frequency is reduced. You can isolate a 1 mutant out of 100 plants. You can isolate 1 mutant out of 1,000 plants or the 10,000 plants. Okay? That is the beauty of the. Suppose I have taken a high yielding variety, and as per my objective, suppose I want to do the better nutritional quality, suppose I wanted to do the resistance to the biotic and abiotic stresses. So suppose I have taken a variety, say SD2967, which is very famous in the wheat cultivation. Around 60% area of the wheat is under that variety. But now, suppose if it is suffering from the yellow rust, what is the solution? So that example I will give it to you. So, and generally there is a confusion with the people that whether it's a GMO or not. GMO is a totally different thing. This is manipulation within the genome. It is within the genome. It is not any gene, it's not brought from the outside. So definitely it is. But sometimes people misunderstand things. go back to the little bit of history of the mutation breeding. It's a Hugo de Vries who has coined the term mutation. And the Muller is one person who worked on the Drosophila in 1927, and he got the various mutations, and he got a Nobel Prize for that. And in the crop plant, Stadler is one person who used the X-rays in the barley, maize, and wheat, and from that point onwards, the mutation breeding started. Now, this is just a simple scheme I will tell you. This is the wheat variety with this genome, and we have done the irradiation, and this is how this new gene has appeared, or the modified the genome and the new character has appeared. You get various mutations. Once you start the mutation breeding, you get the unwanted mutations also. Generally, because the mutations are recessive, and therefore they are the sometimes lethal and the undesirable. So you will have to reject such mutations which are not good, and it may lead to the because the frequency is very, very less. So most of the plants will be like a normal type. So that also you reject. And as per your objective, whether it's a high yielding, disease resistance, adaptation, or the better nutrition, okay, you select. So two things in the mutation breeding is very, very important. So that is, one is you have to fix your objective, and then do the mutation, and you should have the, just now I was talking with our head of the department, and she also expressed that screening technique. Screening technique is a very, 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 very important. I will show you in the next topic. So to start with the mutation breeding, what is required is a lethal dose 50. Fixing the lethal dose 50 is very, very important. How to fix the lethal dose? You treat your seeds with the various doses of the gamma rays. There are the different kind of the mutations, physical and the gamma rays, because generally we go for the gamma ray treatment. So fix the dose of the gamma rays and see how the growth is happening. So somewhere, if there is a reduction, 50% reduction, then the control one, that is called as a lethal dose 50 or the GR50. Lethal dose 50, where suppose you have taken the 100 seeds, irradiated, and where the 50% survival is there, so that is called as a lethal dose 50. So upon fixing that one, the various changes occurs at the genetics level, at the chromosomal level, at the gene level, okay, and in the chromosome level, it is like a normal deletion, deficiencies, duplication, and the inversions, translocations, all this is happen. And the gene level, say like a transition and the transversion, okay, changes within the purine and pyrimidine, between the purine and pyrimidine. So that is how the break happens in the DNA, and you get the large number of the variations. This is what the overall protocol of the mutation breeding. So you start with the variety. Say, suppose I have given the example of the SD2967, which is a highest yielding variety and covering a large area. But suppose it has a defect, say like a yellow rust. Yellow rust is a new disease. So now my objective is to isolate the yellow rust resistance in the SD2967. What is the advantage of it? Already it is a high yielding variety, and I want to rectify the one particular character. Say, now you people will use the CRISPR, but I will go for the mutation. The difference is because you have the gene in your hand, and that you will manipulate with the CRISPR, okay? But the way again the reports are coming, we do not know with the CRISPR again it will be precise or not, okay? I also don't know, because I am doing the hitting my seed in a dark, okay, bombarding, taking the pistol and hitting in the dark. I do not know whether the mutations happened or not, okay. So what I do, I treat the large number of the seed in the M1 and grow the large population, say like a 50,000, say like a 1 lakh, okay. I will grow this population in the area where there is an endemic situation for the yellow rust. What I said? Screening technique. Unless and until you fix the screening technique, whether it's a biochemical analysis, whether it is a field analysis or any character. Unless and until you fix that one and standardize your screening technique, then and there is possibility. And in that case, I will say, you dream the character. You select a high-yielding variety which is popular, and you dream the character which you want to modify, but you standardize your screening technique. Your dream will be fulfilled. Your dream will be fulfilled. I will give some of the examples for that. So, in the M2 population, when you grow the large, in the M2 generation, when you grow the large populations, this is the kind of some segregation you will get. This is just an example for the chlorophyll mutations. This is the normal type green and this is the yellow. Okay? This is the lethal mutations. Likewise, in the Mendelian fashion, suppose a one particular plant, say I have taken the 10,000 seed, irradiated, and I have grown their plant to row progeny in the M1 generation. Okay? 10,000 plants plant in a M1 generation. And when I harvested it and grown the 10,000 plant to row progenies, maybe one of the plant progenies will segregate for the particular character. Okay? So, this type of this. Now, this, because it is a mutation, because it is a random, so I do not know in which plant there is a mutation. So, unless and until I fix my screening technique, say with yellow rust. So, what we have done? We have put this in the BISA campus where we have done the artificial inoculation also, and there is a natural condition also. So, almost all plants, because the original variety is susceptible, they have shown, and there were only 5 to 6 lines which were segregating for the resistance and the susceptible. So, job is done. The population was like a 50,000, 60,000 plants. Okay? So, within a year or within a 2 year, we could isolate that particular mutations. Okay? So, when otherwise in our traditional breeding program, you first of all screen and then you try to transfer that character to the high-yielding varieties with the backcrosses program, and then you develop. Now, within 2 years, we had a mutations. Now, we are evaluating that one. It is, some of the mutants are as good as the SD2967, and some of the mutants are better than that. Some of the mutations are better than that. Okay? Because the mutations, it is not only for that character. Though we emphasized, though we fixed our objective for the yellow rust, but because it is a random process, so it has given the mutations for the other characters too. It is hitting in the genome. So, simultaneously, we can improve the large number of the character. But do not dilute your efforts for the other character. You fix the character, first of all, isolate that your mutant, and beside that, because it is a hitting in the genome, you will get the large number of the population. So, you can isolate that also. So, this is how you have to fix your objective, fix the varieties, okay? Standard screening technique you follow and you isolate the mutations. I was talking with Dr. Ranjit now. We have given one project to the BCKV and now in the cotton HTBT is coming, okay? Herbicide tolerance also. So, we thought that why not to go ahead, okay? And in the lentil last year, in the M2 population, the population must be more than 2 lakhs population. And out of that, 2 progenies we got the herbicide tolerance, okay? And one of the very good example in the IRI, Dr. Ashok is there, he has developed the rice. That is also through the mutation. Then he transferred to the high-yielding varieties, okay? So, our generally we say, as I said, that you dream the character, fix the high-yielding varieties, and then fix the standard screening technique. There is a likelihood of getting your mutations more than 99%. That is all. And ultimately, this is what the fixation of the mutations, breeding, and then evaluations, and the release as variety. Okay. What are the characters? As I said, I have given the example of the disease resistance. You can go for the yield and incontributing characters, morphological character, flowering, early flowering nowadays. Okay. the pulse varieties which are coming 60 days with the high yielding potentiality. Okay. Plant type. This is what in the future it is going to come as a one of the aspective, though it is a very old concept of the 1966 which are given by the Donald in the wheat. But now, because of the shrinkage of the resources and the bombarding of the new problems with the climate change, we will have to develop a plant which will sustain under such adverse conditions. So that is what again the major objective. Then resistance to shattering, resistance to biotic and abiotic stresses, seed dormancy, anything, in oil quality or the protein adaptability, biochemical pathways, whatever nutritional. Ma'am was saying that now we should develop a rice variety which will we will eat that carbohydrate, okay, or the starch, but will not have that glycemic index. Okay. So these are the challenges. Now because the people are, now we will have to work accordingly. So in my opinion, mutation is the best option for that. You can go for the CRISPR, you can go for the tilling, you can work for the molecular, but develop first of all that kind of the variability. That is possible through the mutations. Now I will give you some examples. Okay. As I said, you dream the character and you develop the mutations. Now just I will rush through some of the slides. You see the flower color, you see the leaf shape, seed type. This is the brassica again. You see the number of the packed seed. This is what the standard control, which gives the 12 or 13 seeds standard, and here we have near about 21 seeds, but the silica is small. Okay. If you increase the number of the seeds, maintain the number of the silica or increase the number, your yield is likely to increase. Okay. Seed color, if you see, any kind of the seed color, okay. This is what our parent, and we isolated, and we have released the varieties with the collaboration with the PDK via Cola. This is the first variety of the India, Surya. Surya is the sunflower is not the cultivated crop. It was introduced somewhere in the 1960s. But this is what the germplasm we got. Though it was a high yielding, it was not preferred in the market because the oil content was less. The preference is for the black seeded. So maintaining the yield potentially of the Surya, we have isolated a black seeded mutant. See, this is the standard flower and the variability which you got in the sunflower, flower color. This is one of the dwarf mutant which you isolated and characterized, and now possibly in the this year or the next year we are going to put in the yield trial. You see, this is the Varuna high yielding varieties, very highly adapted varieties. Okay, but if you see, this much portion is used in the reproductive phase, but most of this portion it is going as a waste as a energy, though the biomass is good. What we have developed, a dwarf plant, okay, where the vegetative phase is not much, but the yield potentiality of this is as good as a Varuna. If you look into this, there are the two plants, you see the number of the bearings of the silica. So that is how we have to, as I said, tailor the plant type. Okay. Now with the minimum resources and the agronomic practices, because this is dwarf early, it will exhaust, it will try to utilize maximum nutrients within the short period, and it will give the same yield. Now this is going to be entered in our All India coordinated trial. This is the early linseed. This is one of with the climate change, the farmers are facing the problem. It's our crop is ready, but suddenly suppose there is, even last year I think there is a lot of loss of the wheat, and if this happens when there is no sources for the resistance, for this is the pre-harvest susceptible, if the rain comes, your total wheat is gone. But suppose you develop the mutant, okay, which will be the pre-harvest sprouting resistance. So we have isolated, even in the same mutant population of the ESD 2967, so we have isolated the pre-harvest sprouting resistant also. Okay. This was the example I was giving for the yellow rust. This was tested in the Bisa in the Punjab, and we found that our mutant was resistant, clear cut, No, I will say the immune type. Sometimes even we say the resistance grade 1, grade 2, though we have that kind of the mutations, but some of the plants were immune, even not a single spot of the yellow rust on the plant. So that kind of the mutations. I was talking about the herbicide. We have conducted the experiment on the herbicide also, sprayed where most of the plants are died. Okay? But we got a few plants which shows and this is to be confirmed. This is just, it is not this is just for the information. We want to confirm this in the subsequent generations. With the drought situation, the level of the water is going down and down. So if you develop the plant which will have the long rooting system, so there is a possibility of the survival of the plant even in the drought situation. So these are the examples with the other crops also. So using this kind of the mutations, I can show you the large number of the pictures, but to cut short the stories, I have put some of the specific examples. So it is not only the BARC or in India we are using the mutation breeding. Mutation breeding is being used all over the world, and using this kind of the technique, so far 3,250 mutant varieties have been developed in the 175 crop species and the 60 countries and the 91 years of the efforts. These are the list of the various countries, how the China is topping, followed by the India, and then the Japan and the various countries. This is what the percent contribution. The maximum use of the mutation breeding has done in our continent, that is Asia. This is again the one specific example I will give at the international level. In the Japan, say 50 years before 60, there was a devastating disease called as a black spot disease in the pears, on the pears. And almost all cultivated varieties, all the plants in the Japan, they suffered a setback of the black spot disease. But during that time, what they found, there was one plant in their gamma field. Okay? There was a gamma field in the IR also, but nowadays it is. So they cultivated fruit trees and the other crops. The gamma field is a area where they keep the source of the gamma source, okay, cobalt-60 source, and every day they will expose the plants, okay. So when the energy comes or the rays comes, they will hit to the growing buds and there will be the mutations. So during that situation, what happened, there was only one plant of the pears and there was only one branch which was not suffering from the black spot disease, okay. So what they have done, they have isolated that and then they have vegetatively propagated, and the present day cultivation of the pears, now Japan is the one country where the lot of exports of the pears it is out of only that particular branch. There are the two varieties they have produced out of that, okay, which is resistance and present cultivation. So that kind of the miracle can happen this is another miracle, I will say. The world was worried about the UG99 disease in the wheat, okay, which was first appeared in the Uganda, and it was likely to spread to the all over country. And there was a big measure was taken even by Indian government. Germplasm was sent to that area, then they screened and all this thing, and we have now but there was no solution. So what the peoples have done, Miriami Kiriana, and he is a Kenyan plant breeder, he initiated a mutation breeding program. He has taken a 10 kg seed of the 5 varieties. Now just to imagine 10 kg seed of the 5 varieties, the number of the seeds, total number of the seeds. What I was saying, you grow the large M2 population unless and until your M2 population is very, very large, because your frequency is, say, suppose 1 out of 100 or 1 out of 1,000 or 1 out of 1 lakh. With that proportion, you have to grow your population and screen under the stringent condition. That is what, again, I want to emphasize, your standard screening technique. Okay? So they have grown that, maybe the 5 hectare or more than that area in the M2 population, and then they have isolated a 7 mutant out of that. And now 2 got released. And there were large number of the seminars, symposiums, meetings, policies over the UG99. And the solution came out of the mutation. Okay? So when the doors are closed in the crop improvement program, I will say that start mutation breeding. And these are the examples. It is not because I am working in the Bhabha Atomic Research Center and therefore I want to emphasize. No. This is the way that when there is no solutions, so only way one can come out through the mutation breeding. Vietnam, once upon he was importing the millions of the rice for their survival. And when they started the mutation breeding in 2007, they have started the exporting the rice. That is how examples. So like this, I can give a large number of the examples of the success stories of the Japan, Italy, okay, China. China is the one of the varieties. He is the first to develop the near about 700, 800 high-yielding varieties in almost all crops. In the once I visited to their Chinese research station in the rice, they have showed me a plant which was only single come. Okay? At that time, first time they reported that was a mutation. So they were doing all molecular biology of that. Through the mutations, there is a solution for the existing problem, and at the same time, you can develop the material for the basic studies to understand the plant. Okay? Now, once you say the rice, there are a large number of the tillers. Suppose I wanted to study what is the phenomena of the tillers. What are the genes responsible for the tillers? So, unless and until I have the contrasting character for that, then and then I will be able. You see the Arabidopsis mutations, why the genomics is successful or why the sequences was successful in the Arabidopsis, because they isolated large number of them. You name any character, they have the mutations for that. So in the genetics or the breeding or in the genomics, unless and until you have the contrasting character, then and then you will initiate the genetic studies. Then and then you will initiate the biochemical studies, characterization or anything. Okay? So beside our big success of the achieving the particular objective, you can generate the large population for the basic studies also. I have developed within the same population of the sunflower, I have developed a plant which was just 10 centimeter tall. Just 10 centimeter. I have not shown the picture of that. Just 10 centimeter. Okay? But I have not done any detailed analysis of that. So what I meant to say, there is a possibility of isolating the large number of the character. And in the USA also, they have done. This is the drought tolerant in the Cuba country, tomato they have developed. So this, you see, this is the first released varieties in India using the different mutagens and the crops. And again, it is classified as per the various crop plants. Okay, the number of the mutants now must be 345 or so. And the character is a high yielding, early maturity, disease resistance, quality, grain quality, abiotic stress, improved plant type, and the other characters. Okay. This I will give the example of IRA itself only. In the chickpea, these are the varieties developed through the mutation breeding, and they were so popular. Their breeder seed production as well as the certified seed production, it was really phenomenal. So this is how the mutations can help even in the increasing. In the Bhabha Atomic Research Centers, we have developed near about 46 high yielding varieties in the oilseeds, pulses and the cereals. I will just go through some of the examples like groundnut. We have done I will say the miracle, our TAG24. It's a drought tolerant variety used in the summer cultivations, and now it is being used as a check variety. So there are the two possibilities. When you have taken the high yielding varieties, inducted the mutations, isolate the mutations, the same mutant can go as a variety, or if it is not giving that high yield, what you can do, use that mutant in the crossbreeding program. So that is called as a cross derivatives of the mutant variety. Okay, they are the two channels. Okay, so our example, some of the some of the varieties are the direct mutant, and if you use in the crossing program, that is called as indirect mutant varieties. Okay, so using both the approach, we have developed these varieties in the various crops. This is a list of the this is the TG37, very bold seeded, 41, and TAG24. It's a way. And if you see the, we have almost covered the all India for this. These are our popular varieties in the various states, almost all over India. Again, our bold seeded varieties. And this is how the farmers are happy in the various states to cultivate our varieties. Now, I will say you can see near about 10 to 11% of the breeder seed indent. Now, I will say in India, there are how many? 52 agriculture universities, ICR institutions, and it's a big force. And suppose I say BRC, there is only one person, or at the most 2 persons working on the particular crop. But we feel happy that whatever may be the means, whatever varieties we have released, so their indent is say out of 100, suppose BRC is contributing the 10%, by the 2%, I will say it is a great achievement for us. And I will give one example of the farmer. So here in the Yavatmal, we used to go regularly. So 2 years he has cultivated, see likewise there are large stories. It is not for each and every university has the success stories. Now it has put it in the ICR site also, I think 2 days before, the success stories of the crop improvement, and the crores of rupees has earned by the ICR by developing the high yielding varieties. So that is there. But from the BRC side you may not be knowing, so therefore I am saying. So we used to go, and third year when we went by that road, so we confused in the where is the location, where is that, and we were searching, no, somewhere it was here, because where it was a small hut, it was a big building. So then the farmer came out and they said, no sir, यही है वो घर, हमारा है भाई. तो उन्होंने बोला ये तो भाई बिल्डिंग होगी. सर बोले ये ग्राउंडनट की वजह से हुई है, 2 साल हमने कल्टिवेट किया है. द बेस्ट ऑन दैट बेनिफिट, इट इज नॉट जस्ट टू पोज, बट the unless and until, this is just a message, unless and until we work for the, unless and until we work for the improving the economy of the farmer, then and then whatever problem all over India we face, then and then there is a possibility of the, we'll get our payment, we'll get our salary, we'll finish our job, we'll publish the paper, but unless and until we reach to the farmers at this level to improve their economy, then and then it will be a big success for us and ultimately for the people. This is how the groundnut field of the progressive farmer. See, this is number of the minikit trial we have conducted all over India for the various varieties in the almost all states. This recently, 2009, now ICR has notified this variety TL99, and there was a big news in the Indian paper, and the ICR has given this news that now we have developed the linseed crop as an edible oil crop. Linseed is not edible oil because of the high amount of the linolenic acid, okay, 50%, it's not edible. It was used for the industry purpose. That we reduced down to the 0 level and developed the varieties called as the TL99, and this year it is notified in the 2019, and it is a big news for the country. So therefore, we feel satisfaction for that. This is the mustard variety. BRC is the first variety to develop the yellow seed coat mutant. In the 1960, the mutant was isolated, and the same mutant, when I have taken over, we have released as a variety, okay, and that is the first yellow seed. Now, what is the advantage of the yellow seed coat mutant? It is having the high oil content. The oil color is appealing. You need not to do the decolorization in the industries, and therefore now in the 00, they plus yellow seed is they called as a 000. Okay, so BRC is the first institution in the country to develop, and that particular mutant was extensively used all over India to develop the yellow seed coat mustard in India. Another feature of this, when that time it was just the yellow seed coat, but when I have analyzed recently, it has the reduction, 50% reduction in the erucic acid. In the mustard, they are having it is 48 to 50% erucic acid, and this mutant is having the reduction in but that time we didn't analyze. So that is again the beauty. Okay? When we select for the one particular character, there could be mutations for the other characters also. Now, beside the yellow seed, it is early, erucic acid less, little bit of dwarf plant type. So, when you say, now suppose I say in a one mutation, there are the 2-3 mutations occurred. Okay? So, in the standard breeding program, we say as a pleiotropic effect. Okay? But we do not know whether it is now with the genomics. Suppose I say there must be some regulatory factor, major regulatory factor for that, and the genes are aligned in the sequence, and probably there is a mutations in that. So now with that, I have the material to go for the genomics of this particular. So whether it is the same gene for the or regulatory for the other genes or not. So you will be able to publish a good quality papers also, which will publish in a very good journal. Okay? So that is also another help. This is again our new variety of the mustard, which has been released for the West Bengal now, TBM 204 with the yellow seeded mustard. And now these 4 varieties are in the pipeline for the various characters, and probably this year again. In the pulses, again we have the good success in the mung bean, particularly yellow mosaic viruses in the black gram and the green gram. These are our varieties. But this particular variety, TMB37, TRM1 and TRM2, it's a powdery mildew resistant. Okay? And this even in the, they have identified in the PAU last year or last year. But again, it was, I think there was some kind of the race specific problem in for the yellow mosaic virus, and therefore I don't know at present situation. But the yield potentiality of this variety for the various environment, we have tested in the West Bengal, there is also it is preferred, Punjab, Maharashtra, and so many states. So now with the climate change, we say the adaptation of the variety. So if you can develop a mutant or the variety which will have the wider adaptability, and then it will be very good for them. Among the tour, this TJT 501, if you go to the Chhattisgarh area or the MP now, okay? 2 years before we went, hectares together on the both side of the road, hectares together. And the farmers are very happy to cultivate that variety. So it's just an example of the various varieties cultivating the TMB37 by farmers. TAU1, we used to say that suppose we have eaten a idli or the dosa today, it is out of that because the 90% cultivated area of the Maharashtra urud bean cultivation is covered by this area. That we can see through the breeder seed production and the certified seed productions. These are some of the varieties for the what will say the after rice they cut, and in the rice stubblings are there, they broadcast it. Okay, that this is the Uttara conditions. So some of the varieties. This is the 501 in the MP area, very famous. Again, in the coming to the BARC contribution in the pulses, again it comes near about 8 to 10% contributions by our breeder seed production. And this is how all over India we try to see that how the peaceful use of the nuclear energy in the field of agriculture has reached to the farmers and the various states of the India. This is just out of this thing. This is not a crop plant, but this is a green manure crop. We have developed a mutant which is having it's a Susbenia rostrata, and which bears the nodules. But what we have done, it is a photoperiod sensitive. We have isolated a mutant, it's a photoperiod insensitive, so that it can cultivate throughout the area, throughout the year, okay? Which fixes the nitrogen, the stem nodules, and it produces the near about 120 kg of the nitrogen in a 55 days or the 50 days. And with the what to say, the level of the soil or the fertility level of the soil is coming down, I think that this is, though there are the other varieties, but now we are trying, we are taking the trial in the various universities, and the first year trial they are showing very good results out of this mutant. Okay. So this is how, this is the photographs of the Homi Jahangir Bhabha, who is the father of the nuclear program in the India, and this is out of the various seeds, and this was presented in the Vigyan Bhavan 5 years before, when there was a international symposium on the nuclear sciences. Now coming to the another technique of the mutagenesis is the transposable elements. It is not that we are, when you, now if you say the genome as such, say 50% is active and 50% is a inactive basis. And so far there was no, what you say, the attention towards the 50% of the, only we were concentrating on the active genome. But 50% genome is a treasure for, now they say that it's a treasure for the crop improvement. See, in the evolution it is so happened that nature has given us something, but it has kept a treasure for us to exploit nowadays. In the adverse condition, if you see the evolution, in the adverse condition, the plant adjusts that adverse condition by taking the genes from this treasure. They activate themselves on the adverse condition, they fish out the gene from this, and then they adjust with and that is how the evolution has taken place. So if you activate the transposable elements, so some of the things which is not possible through the standard mutation breeding program, there is a possibility to isolate that variability. I'll give an example. This paper has appeared in the Nature Review Genetics. This is what the parent, with the activation of the insertion of the transposable element, this has become green, and the recombination of the same thing, it has become a red. Otherwise, it's very difficult sometimes to get this kind of the variability through the standard breeding program. So if you do, and this, if you want to go through, this is a very good paper, and see, therefore I was saying that with your development of the varieties, you will get the novel germplasm, but if you analyze this in such a way, your paper will be published in a very high impact factor journal. This is how, again, this is generally from the, see, previously we have seen from purple color we can come to this stage, but we cannot go reverse. Reverse in the sense, suppose this is in the pigmentation pathway, we can say from black we can come to the white with the range of the color in between. But from the white we cannot go to the black. So that is what our understanding so far. The same thing happened here also. From yellow, with the insertion of the transposable elements, they could go to the reverse side. Okay? And they have isolated even the black red type or the dark red type of the mutants out of that. This is the tilling technique you know very well, and if you have the gene in your hand, you can do the precise mutations. And previously, which you say the character which is governed by one gene. Nowadays, there are the large number of the alleles even for the single gene which previously used to say. So, and it is being done in the so many crops. This is one of the example I was telling. When you take this is the parent plant and this is the mutant plant, and when they crossed it, I was saying one is a direct mutant, and the second approach is take a mutant in a hand and then you cross it and try to develop the high-yielding varieties. You see the amount of the heterosis they got it, which otherwise if you take a standard germplasm, probably you may end up with the 10%, 20%, 25% increase. This is, you see, near about maybe the 100% increase of that. That is the beauty. So whatever mutant you got it, you don't throw. Though I have seen in the first type, negative type of the mutations, even though if you isolate that mutant, don't throw. Keep as a reserve. That can go for the basic genetic studies. And in the, suppose you have the 10 mutants, try to cross it with the high-yielding varieties, try to cross it, see the combining ability. Probably one of the mutant may end up with like this. This was they were studying the florism pathway, and they wanted to study that particular gene and the pathway for that. But in the process, they got this kind of the mutations. Though this is how the breakthrough can happen. Now the CRISPR is again the one of the technique now, and large number of the characters in the various croplands now they are being modified, and this is the powdery mildew resistant. This is the again the what will say the ripening gene, and large number of the characters in that. And now I will give you some example for the nutritional aspect. This is what the essential 14 known nutrients that is required for the human being. But again, if you see the report, there is the suffering of the people because of the nutritional deficiency. Sometimes if you look into the data, it is really feel that whether now you are sitting, I am talking here, but our health would have been something different had it been analyzed in our childhood and provided our parents would have given that particular kind of the nutritious food. But even with the education and all this and the changing environment, now if you see though we are educated, okay, we have the kind of the information, but still, really we are taking care of our kids in that way, nutritional point of view, okay? Analyzing that one, going to the hospital, no, what kind of this thing, no, still we are going routine way. Though, unlike the previous year, now we are trying to give our best. But in the world, the situation, and probably if you see the Indian map, so you see this many children in the India. Still, this is again the 2018 report. Okay, so there's a lot of this. Suppose this kind of the generation we are producing, it is not you and me. Large number of the mass in India is suffering. This again the report, if you see the 53% of the adult women and 50% of the pregnant, they are anemic. So if you say the 50% people, 50% our generation, if the woman is anemic, what kind of one and other kind of the deficient this thing, probably it is noticeable, sometimes it is not noticeable, or throughout his life period, somewhere it will express. So I think there is a need, and this is very good that this division and Dr. Ranjit has arranged this training program. In the future, at least, you will notice this and try to develop a cropland, improve the croplands on the nutritional point of view. Again, this is the map showing this is the terrific situation for India. Now, biofortifications can come through the agronomy, transgenic, and the breeding method. And there is a large, if you see go to the site of the Harvest Plus in Australia, they have used the mutation breeding program extensively. And only through the mutation breeding program, they have developed the biofortified crops. And that germplasm is being supplied to the all over country, including India, and some of the biofortified varieties are now coming up. So I need not this might have already been stressed in your, but this is how you see the zinc-rich mutant China has developed. And if you see the various, including the mutation breeding, okay, standard conventional breeding with the mutation breeding, these are all the biofortified crops for the particular nutrient. And again, this is the LPA, low phytic acid mutant in the various croplands. These are the three peoples, they got the World Food Prize by developing a mutant variety of the sweet potato, which is having the higher amount of the vitamins, all this. So there is no need of worrying that suppose I am working on the plant breeding, it will take a long time and probably I may not get any award. So don't worry about that. You start mutation breeding and probably you will be the first person to get some kind of the awards too, besides your publications. So there is a good scope in the mutation breeding. This is again the QPA protein. And this is what the Harvest Plus program in the large number of the crop, particularly cereals and the biofortified character country. And this is what the agronomic trait. Again, as I said in the oil seed, oil quality can be improved. Now, I was saying one of the mutant which has shown the 25%, 50% reduction. Recently, the dwarf mutant which I isolated, that is also 25% reduction. Now, for the erucic acid, there are two genes, E1, E2, E2. Okay, two genes are there. Now, suppose these suppose I have the two mutant, and if these mutants are for the two different genes, one is for the E1, one is for the E2, probably we have initiated that study, and now we are screening the F2 population, and probably if it is a different mutations, we will isolate a zero erucic acid mutant. And if it is so, that will be so far there is only one source in the world for the zero erucic acid, and probably ours will be the novel source of the erucic acid, zero erucic acid. So that kind of the change. So in almost all oilseed crop, one can do we have isolated a high oleic acid mutant in the groundnut also. So this is how the Borlaug says you cannot build a peaceful world on the empty stomachs and the human miseries. That is correct. And we are here because of the farmer. My humble request to all of you that while eating, see that your food is not wasted. Thank you very much.
So now the talk is open for the discussion. If you have any queries or questions, please come in front.
Yeah.
Thank you, sir. Thank you for wonderful talk. I want to know about how insect and pest are controlled by nuclear energy.
Oh, okay, that is not our topic, but there is a technique called the sterile insect technique, SIT. Okay, so what we take in that system, we take a males, irradiate it. Okay, so because of the irradiation, there will be change in the fertility status. The fertility status come down, and when you cross it with the female, the population which will be generated, it will be sterile. Okay. And in the subsequent generation, that sterility from the 100% come down to the 0%. And that is how, but what happens in the open field, it is not possible. So suppose we have the island, we have worked on the this coconut weevil, where the population will be always met within that. So then there is a possibility. But in the open field it becomes, but now we have the very good program on the SIT for the so many insects.
So one more question, the different mutagens that we have used, they have any effect on metabolic pathway?
Yeah. See, any mutant, mutagen which is used, see, as I said, we are hitting in the dark. Okay, we do not know what kind of but any kind of the phenotypic results, it is because of the change in the biochemical pathway, it is change in the physiological pathway, if you go into the details of that. So there is a mutations for that also.
Thank you, sir.
Yeah. Yeah, please.
Thank you, sir, for this informative lecture. So, in my UG course, I studied the most important disadvantages for molecular breeding is the that the most mutations are recessive. So their expressions are not visual due to the dominant allele. And the second is the mutations produce through the molecular breeding, mutations produce the pleiotropic effects, and it hinders the multivariate evolution. So what are the strategies to address these issues?
Yeah.
During the screening technique.
Yeah, so you are correct that the generally mutations are recessive. Generally the mutations are harmful. Okay, what I try to emphasize, I was emphasizing your screening technique. Okay, so you screen the mutations which is useful for you. Okay, and the recessive mutations I have shown even in the slide also called as the negative mutations. Okay, you reject that. But when I have given the example of the tomato, what I said that you keep all kind of the variability. Okay, we do not know in what way it is useful in the crop improvement. So try to cross it and see that maybe some of the mutants may surpass the expected heterosis. Okay? So in that way that can be used. Now for the molecular breeding, as you said, even the TPM one which I have shown, I said that probably 3 characters has mutated. Okay? So probably it could be the pleiotropic effect also. But when you want to study the molecular aspect, what you do, when you isolate a mutant, okay, immediately you cross it with the back cross with the parent. Okay? And that is called as a mutated near isogenic line. Say reels when you produce it, so nils you produce. So likewise, when you got a mutant, you backcross with the parent, and in that way you are near to the parent for that particular gene. So you can avoid the another effect or the pleiotropic effect of that particular thing. Okay, thank you.
Please.
Yeah.
Thank you, sir. Thank you, sir, for enlightening talk. Sir, my question is that up to what extent it is better than this mutation breeding is better than CRISPR-Cas technique? Because in CRISPR-Cas technique we directly hit on a target site, and in mutation breeding there is
We hit on the genome. Yeah, you are correct. Even before saying you, I said we are hitting in the dark. But unless and until you standardize your screening technique, be precise for your objectives and isolate only that kind of the mutation. See, any technology, if you do, there is the advantage, there is a disadvantage. Now, if you see some of the CRISPR paper also, in the mustard, I have seen one paper, their objective was something different, but they could isolate the dwarf. Okay? So, there also that chances is there, because even though nowadays we say that we have the specific gene sequence, but we do not know in a totality how it expresses with the combinations of the other gene. Recently there was one person from the very good company, I will not take a name of the company, they came to me for the seed irradiation. I said, why? And well, you people should do the CRISPR. But it is his opinion that the CRISPR, through the CRISPR is also not that precise. I don't know how far it is correct. There is a possibility of affecting the other character. But I don't want to say anything on the CRISPR, but on the mutations I will say, yes, there is a other gene, affect the other gene, but you should be precise during the screening technique, the character of your importance. That I guess. Yeah, please.
Sir, there is a different mutation techniques like gamma rays and different types of rays and chemical mutations for differences like drought tolerance, biotic and abiotic. So there is a specific treatment for specific track, like if you want to do nutritional enhancement, then there is a specific.
No. See, as I said, we are hitting in the dark. Okay? So we do not know the what kind of the mutations. If you use the EMS, means there will be this kind of the mutation. If you use the gamma rays, means this kind of the mutations. It is not like that. But with the gamma rays, it is the large portion of the chromosomes they get deleted. But in the chemical treatment like sodium azide or the EMS, you get the point mutation, okay, for that particular gene. But screening is important. It is not sure that the specific mutagen is for the specific character. It is not like that. You can isolate any type of the character, which I have given the list of the characters, okay? But it is not a mutagen specific. It is not a mutagen specific. With the screening technique, you can isolate the character of your imports.
Thank you. I think we will take questions later on. So the essence of any agriculture technology is how best it brings a smile on the face of the farmers. And mutation breeding has come a long way. And sir has nicely explained different aspects of mutation breeding and different success stories of mutation breeding. And we are also working in close association with BARC, and we have one project, and then so I think
I'm sorry, I have not mentioned this.
and we are so our objective was to find out some of the mutants which were really responsible for modulating the tolerance of wheat. and we use different omics tool because the title of this training is omics. So let me tell you one more success story. And then we could able to find 8 genes which were really responsible, instrumental in modulating the tolerance of the wheat mutant, which was developed by BARC. Now it is at M7 stage, and hopefully, so maybe in coming years, so it will be also one of the feather in the cap of mutation breeding.
Thank you very much.
So
Thanks a
with
lot.
this brief note, now I request my Dr. Bhupender Singh sir, principal scientist from SISCRA, to please felicitate our today's speaker.
Before I felicitate, let me take this opportunity to tell you all that it is always a treat listening to Dr. Jambhulkar, and he's my senior, but also a very good friend. Okay, it's always a surprise when you see his lecture, and he always has something up his sleeves every time he delivers a lecture. Be it, I just want you guys to take note of the mustard varieties that he developed, where he could reduce the vegetative growth of that, but without altering the yield aspects. This is one example wherein you can think of improving. Why we should have too much of vegetative mass if it is not required. Okay. Second aspect which he, although he gave a lot of success stories, all of which are classical examples of successful use of ionizing radiation for mutation breeding. Second example which I would like you to take note of is the groundnut, which he showed the mutant. And if you had seen it very clearly, the mutant that he showed was selected based on the vegetative mass, because the mutant had a higher vegetative mass, but the groundnut yield of that mutant was less. So then what they did is, then they backcrossed with the parent to get a line which was having not only a optimized vegetative mass, but maximum yield. But selection, groundnut is underground. You cannot select based on the underground. You have to dig it. So without digging, how you can select? So their selection in that case was based on the vegetative mass. If you see the plant, you can go back to this YouTube video and see the plant. That plant was selected, the mutant was selected based on the perfused vegetative mass. Okay, and then they backcrossed it. So it is really, really, you guys, biochemists or breeders, now science has no limit and boundaries. So I really feel very privileged to listen to you, sir. I am sorry for being late. It's a pleasant duty to felicitate. Now I request.
No, before that, I will just say the character which he has explained. Some farmers have said this, which generally I don't say that is because that is not good word. He says, "Miya mudbar dadi hadvar." So when you have developed the groundnut plant, which is giving the more pods, and the vegetative phase is a very, very less. So he denoted like that. It's not good to say, but he says that the Miya Mudbar and Dadi Hadvar means एक ये है, but उसका जो reproductive phase है, it's very, very big. So that is what. Yeah, thank you. Yeah, thank you.
Thank you, sir. Thanks for coming.
And you can note down my email, sjj@barc.gov.com. And if you have likewise, Dr. Ranjit is having the project. We have the financial institution.